You've heard several presenters mention the term GCLP, good clinical laboratory practice. We are fortunate to have available through the core the services of the QA unit run by Marcella Sarzotti Kelsoe. Not only do they keep the people doing the assays in line such that they are GCLP compliant and have greater credibility in the scientific community, but also I think some of the services that Marcella will go over now could be of value to you in consultation as you're looking at assay development or implementation of assays. So Marcella?
Thank you, Kent. Thank you, John, for inviting me to speak. Yes, I'm going to bore you to death with GCLP. I hope not. But some of the speakers already mentioned the terminology. Also, as we like to use a lot of acronyms, we have called our program CFAR GAP. So you probably wonder what that GAP means. So here it's a picture of what it means. G stands for Good Clinical Laboratory Practice Compliant, A for AIDS, and P for Program. This is sort of a comprehensive name for all the labs that perform AIDS studies in compliance with GCLP. This is kind of an older slide, older version of what Kent presented, but it's a comprehensive picture of the program, the CFAR GAP program, which includes, in addition to the immunogenicity endpoint assays, also the operations and the administration portion of the group that play a phenomenal role, and as well as the IT system phenomenal role in keeping us all running and running straight. So why do we need a quality program? This is a $1 million question. So when we test specimen for human clinical trial, it is crucial that we implement quality control measures because we want to assure that we have consistency, integrity, validity, and reproducibility of the test results. So we established, and this is a program that goes back to 2003, was a mandate from the AIDS sponsor. We established an HIV vaccine clinical trial testing laboratory unit in which we implemented good clinical laboratory practice guidelines. And this was really a pioneer effort under Kent's group. And what I want to say here, because it didn't show up in the organogram, we have established at the very beginning of this effort a comprehensive committee called the GCLP committee, which had about 2 members from each lab that helped us understand what the processes were in each lab and helped us design the very first SOPs that became very widely used in the program. So if you ever have to start from scratch, be a very comprehensive group and try to include everybody that really knows the nitty-gritty job details and can really advise you on what is needed. And again, other than that, it's also very crucial for the success of a program like this that management and upper directors really buy into the system. Otherwise, if you don't have a buy-in from management, it's very difficult to establish such a program. So GCLP defines how studies are planned, performed, monitored, recorded, reported, and archived. And we present this in all our trainings. This is 6 very important concepts that we follow through in all our QA life. Who should follow GCLP? Now, a lot of you here do research work, and for research work, it's good to have good practice in the lab, but you really don't have to go through the strict level of assurance that we have for the GCLP lab. GCLP standards apply to laboratories conducting testing of specimen for phase 1, 2, 3 human clinical trial. So some example, and you heard a good introduction from Guido, Celia, and Kelly from all the labs in the CFAR GAP program, include the neutralizing antibody assay, ELISpot, now the ADCC, intracellular cytokine flow cytometry, binding antibody ELISA, binding multiplex assays, as well as the blood processing preparation that we have performed for several, and we are performing for several networks. I'm not going to get into details on that. So this is just a pictorial. It was a review that Stuart Shapiro wrote about the various steps of the clinical trial phases from 1 to 3, BLA, and phase 4. And down there, the three little guys are carrying the GCLP torch. They have to step through this very gray area that is not really clear under which regulation is controlled, because good laboratory practice applies to preclinical studies. Good clinical practice applies to more of the patient-related activities, as well as safety assays conducted in the hospital or at the bedside. But what we do in terms of sort of research endpoint assay is this gray area that is hard to define in the regulatory world. In addition to this, there are various flavors of GCLP out there, especially across the ocean. If you compare the US and the UK GCLP, there are a few differences. And so we put together a workshop, and this is, I'm not going into details, but we came up to an harmonization of the two opinion on GCLP. So it's published, you can see it there. So what are the main GCLP concepts for laboratory operation, and what's the importance of quality? We want to stress that the quality is important from the minute you collect the specimen to the very final data analysis. And so these are all the different elements that we monitor as a quality assurance unit. Personnel training, the facilities, we spend a ton of time on standard operating procedure documentation and document control. We make sure everything is documented in terms of deviation and corrective action, equipment calibration and maintenance, specimen, method qualification and validation. I'll touch on it. It's a long topic. I'm happy to answer any specific question beyond this time. Data integrity routine, we do perform routine audits, of course, and then another effort is spent into document retention and archival. So I just want to spend a couple of slides just to go over the steps that are needed to move from R&D assays to regulated, validated assay that Celia and Guido and Kelly already mentioned. And you may have to go down this road, and we'll be happy to assist you as part of the CFAR core. You probably, some of you already have a very interesting assay. You're using it in an R&D lab. How do you move that into something that can be very relevant for monitoring the efficacy of clinical trial in immune studies? And so you usually have a protocol, and you move that into a standard operating procedure. You have to optimize the assay. You go through a phase of pre-validation. You validate the assay, and then you have to implement it. And we had done quite an exercise under David's CAVD, CAVIMC in implementing the TZMBL neutralizing antibody assay worldwide. We spent a lot of time traveling to all the various sites so that they all would conduct the same type of assay in case a vaccine, which happened to be the Thai vaccine, happened. You also, when you change a few elements of the assay, you have to revalidate. So this is all the very crucial steps. Why do we need method validation? Because the assay need to be demonstrably fit for purpose. And so before you submit a product application to regulatory agencies for a licensure, the assays should be definitely validated. And this is the last concept on this slide, is a very common mistake that people make. They think that having intra and interlaboratory comparisons and have positive and negative control when they run their assay means that their assay is validated. That is not, is a misconception, and it takes a long time to validate an assay. We just went through that with the A3R5. So just a pictorial of what an assay validation is. I like to think of it as a balloon, and you stretch it like a piece of rubber in all direction until it breaks. You don't want it to break. You want to see what are the limits of your assay. And within this limit, we know with a high degree of assurance that the method will produce accurate results. I'm not going to bore you, but these are all the various parameters from the ICH Q2R1 guidelines, as well as the pharmacopoeia that you have to study when you validate an assay. And however, before, if you don't have to go through the stretch of validating an assay, we are here to help you for providing assistance in interpreting applicable regulation to your particular lab or needs. In developing SOP, we have probably 350 SOPs under our hands that we can help you definitely in developing or adopting for your lab. We provide help in assay validation. We provide compliance inspections and audits. And we can focus on equipment and reagent management system. We can come and do a site visit of your lab and tell you what we think you need to do to move on or improve and so forth. I also want to tell you that our activities, as they started with a CFAR gap, have now expanded to a lot of other network or groups here at Duke as well as outside of Duke. And so we have this overarching set of standard operating procedures that apply to all these groups, and we are happy to share them as well. And this is the 4 Musketeer that can bother you with QA requests and regulations. If you have any question, I'll be happy to answer, or after.